Mitosis begins with interphase, where the nucleus compresses, the cell grows, and DNA is replicated. Prophase is when the chromosomes become visible and centrosomes take their place at the corners of the cell. During metaphase, the chromosomes align in the center of the cell. The spindle fibers start to stretch out, anaphase begins, and the spindles pull the chromatids apart and move them toward the poles. In telophase, the nuclei reform and the cell splits into two identical daughter cells after cytokinesis splits the cytoplasm. Interphase occurs before meiosis, where again the DNA is replicated. In prophase 1, the chromosomes find their homologous pairs within the nucleus, and those pairs share genetic information. This is called crossing over. During metaphase 1, the spindle fibers expand to reach the pairs, which have now aligned in the center of the cell. In anaphase 1, the microtubules attach to the homologous pairs and then begin to pull them apart, carrying either chromosome to opposite poles. Telophase 1 begins, and the cell starts to pull apart. The nuclei reform, and cytokinesis splits the cytoplasm, leaving two diploid cells. Meiosis 2 begins with prophase 2, where again the chromosomes become visible. Next is metaphase 2, where much like meiosis 1, the chromosomes align in the cell center and spindle fibers from the centrosomes expand to reach them there. In anaphase 2, the spindles pull the chromatids apart and move them further toward the poles of the cell. Finally, in telophase 2, the cells begin to pull apart, the nuclei again reform, and cytokinesis splits the cytoplasm, leaving 4 haploid cells. Each resulting haploid cell contains a unique genetic makeup and only 23 chromosomes.